Hi class, so this is chapter 12, which will be on an introduction to the nervous system, and this will be the final chapter in this module 3. So this module 3 is a little lighter on the lecture chapters to give you some time to figure out dissections, get into your groups, and learn all those muscles. So here we are in chapter 12, an intro to the nervous system. The nervous system is the master control and communication system of the body. There's 3 overlapping functions. Number 1 are that sensory receptors where we'll monitor changes inside and outside the body. A stimulus is any change detected inside or outside the body, and sensory input is information gathered by various receptors throughout the body. Then what your body does is it will process and interpret those sensory inputs, usually in the brain or spinal cord, and this is called integration. Then the body will dictate a response by activating effector organs or glands, and this is called the motor output, a term for the response. So here we have the sensory input. So for example, you see a glass of water. That sensory input gets sent to your brain. Your brain thinks, man, I'm thirsty. And then it sends out a motor response to the arm that picks up the glass of water so that you can drink it. We divide the nervous system into the central nervous system, which is your brain and spinal cord. This is the integrating and command center of the nervous system. And the peripheral nervous system consists of the nerves extending from your brain and spinal cord, like your cranial nerves, your spinal nerves. The peripheral nerves will all link all regions of the body to your central nervous system. And ganglia are parts in the peripheral nervous system that stand for clusters of neuronal cell bodies. So here we have the central nervous system, we have the brain, spinal cord, and the peripheral nervous system. Cranial nerves, spinal nerves, and the ganglia are the clusters of cell bodies. Cranial nerves come off the brain, and spinal nerves come off the spinal cord. So sensory input and motor output. Sensory or afferent signals are picked up by sensory receptors, which are carried by nerve fibers of your peripheral nervous system to the central nervous system. And then you have a motor or efferent signal that carries signals away from your central nervous system. And these motor signals will innervate all your muscles and glands. We can divide your sensory and motor input according to the region they serve, the somatic body region or the visceral body region. Somatic usually has to do with skeletal muscles, and visceral body region has to do with all your viscera or your internal organs. This results in 4 main subdivisions. We get somatic sensory, visceral sensory, somatic motor, and then the visceral motor, which is also known as the autonomic nervous system, or ANS sometimes called. So here we have the central nervous system, brain and spinal cord, integrative and control center. Then we have the peripheral nervous system, we have the sensory and motor division. The sensory things can come from somatic and visceral sensory nerve fibers, which will conduct those impulses to the central nervous system to initiate some sort of motor response, which is conducted via motor nerve fibers. This will conduct impulses from the brain and spinal cord to all of your effectors, whether those are muscles and glands. So we get a somatic nervous system, which is the voluntary going to skeletal muscles, or the autonomic nervous system, which goes to all your visceral, motor, and involuntary muscles. So we have it going to sympathetic or parasympathetic after we divide the autonomic nervous system into 2. The sympathetic division mobilizes your body systems during activity, and the parasympathetic division conserves energy and promotes housekeeping functions like rest and digest. So somatic sensory is the general somatic senses. There are receptors that are spread throughout the outer tube of the body, like touch, pain, vibration, pressure, temperature. And these general somatic senses indicate that they are widespread. We have proprioceptive senses, which detect stretch in tendons and muscles. Proprioceptives is the body's sense of position and movement of each other body part in space. And then we have special somatic senses, which are hearing, balance, and vision, which we will cover in chapter 16. The visceral sensory, the general visceral senses are stretch, pain, temperature, nausea, and hunger. They are widely felt in your digestive and urinary tracts and reproductive organs. And you have special visceral senses like taste and smell. Then the somatic motor response are all the general somatic motor signals that are causing contraction of your skeletal muscles. So these are under voluntary control. So we often call the somatic motor division the voluntary nervous system. And then the visceral motor regulates the contraction of smooth and cardiac muscle. It makes up our autonomic nervous system and it controls the function of visceral organs. It's often called our involuntary nervous system, and we'll cover this in week 4 in chapter 5. The cells of nervous tissue are densely packed, they're intertwined. You just have 2 main cell types, the neurons, which will transmit the electrical signals, and then all of the support cells. They're called neuroglial cells in your central nervous system. They are non-excitable, so they can't transmit an electrical response, but they will surround and wrap the neurons and be support cells for those neurons. So the neurons are the basic structural unit of the nervous system. You have specialized cells that conduct the electrical impulses along the plasma membrane, and this is what a nerve impulse, also known as an action potential, is. They have special characteristics. They have a longevity. They can live and function for up to a lifetime. They do not divide. Fetal neurons lose their ability to undergo mitosis, so neural stem cells are an exception to that. They have a very high metabolic rate. They require an abundant amount of oxygen and glucose, and neurons will die after about 5 minutes without oxygen. So the cell body or soma of the neuron contains all the usual organelles plus some other special structures. The nissal bodies are chromatophilic bodies, which are clusters of rough ER and free ribosomes, and they function to renew the membranes of the neuron. The neurofibrils are bundles of intermediate filaments. They form a network between chromatophilic bodies. And most neuronal cell bodies are located within the central nervous system. And if we have neuronal cell bodies in the peripheral nervous system, if you remember, they are what we called the ganglia. So here's a structure of a typical neuron. We have the cell body, and then we have the dendrites, which are the receptive regions to other stimuli. Here's the nucleus, the nucleolus, the rough nissal bodies. The axon hillock is the area that connects the cell body to the axon. And then here's the axon, the impulse generating and conducting region. And you can see the direction of the impulse always travels down the axon till to the end, the terminal boutons. And there's little spaces. So each one of these kind of circles, blue circles, is called a Schwann cell. And the spaces in between is called a myelin sheath gap or a node of Ranvier. And we'll talk about why those are important. And here's a look at an electromicrograph of a real neuron. The dendrites are extensively branching from the cell body. They transmit electrical signals toward the cell body, and the chromatophilic substances extend only into the basal part of the dendrites and to the base of the axon hillock, and they will function as receptive sites for receiving signals from other neurons. Axons, a neuron only has one axon. It's an impulse generator and conductor of these electrical signals and action potentials. It always transmits impulses away from the cell body. It does not have any chromatophilic bodies, and there's no protein synthesis in the axon. It does have neurofilaments, actin filaments, and microtubules, which provide strength along the length of the axon, which will aid in transport of substances to and from the cell body, and this process is called axonal transport. The axons will eventually branch along their length, but the branches are infrequent until it comes to the end, which we call axon terminals. There's multiple branches at the end of the axon, and we call these branches terminal arborization or telodendria, and they end in knobs called terminal boutons or axon terminals. So then the nerve impulse is generated at the initial segment of the axon at the axon hillock, and then it's conducted along the axon to the terminal boutons where neurotransmitters are released from the vesicles at the terminal boutons. A synapse is a site at which neurons will communicate with another neuron or a muscle fiber cell that we learned in the last chapter. Most information is passed through chemical messengers, and some information is transmitted electrically through gap junctions. So this is a great animation on the synapse between neurons. So go ahead and download that and watch it. We can have 2 different types of neurons. The presynaptic neuron conducts the signal towards the synapse or the junction, and the postsynaptic neuron transmits the electrical activity away from the synapse. And here we have the presynaptic neuron coming down. Here's the terminal bouton at the synapse. You can see how it branches, and it will conduct its signal on to the next neuron, which is located there. So we have the presynaptic neuron is the first one, and the postsynaptic neuron is after the synapse. We have different types of synapses. It can be axodendritic, which is between axon terminals of one neuron and dendrites of another. And this is the most common type of synapse. We can have axosomatic synapses between axons and neuronal cell bodies. So we have the structure of an axodendritic synapse. We have the terminal bouton, the synaptic vesicles, which are carrying those neurotransmitters. The synaptic vesicles will release the neurotransmitters across the cleft to bind to the postsynaptic dendrite. A synapse then is just a very elaborate cell junction. Synaptic vesicles are on the presynaptic side. Membrane-bound sacs will contain neurotransmitters. Mitochondria are very abundant in the axon terminals, and then the synaptic cleft is just the space that separates the plasma membrane of the two neurons. We have different classes of neurons. A multipolar can possess more than two processes, where there are numerous dendrites in one axon. Bipolar possesses two processes. This is more rare. We'll find it in your eyes or other sensory special organs. And then unipolar possess one short single process. The most common type of neuron in the body is the multipolar neuron. So here we have multipolar, what that looks like. The bipolar, two processes, and then the unipolar is one process. The relative abundance and location in the body. So again, the multipolar is the most abundant. It's the major type in the central nervous system. We'll find bipolar neurons in special sensory organs like olfactory mucosa, the eye and the ear. And then we'll find unipolar neurons in the peripheral nervous system, common only in the dorsal root ganglia of the spinal cord and sensory ganglia of spinal nerves. Then the functional classification of neurons, we functionally classify them according to the direction that the nerve impulse travels relative to the central nervous system. So we have sensory neurons that take impulses to the central nervous system. Motor neurons take impulses away from the central nervous system, and interneurons connect the two. So sensory neurons transmit impulses toward the central nervous system. All of them are unipolar. The cell bodies in the ganglia are outside the central nervous system. They have short single processes that divide into a central process, which terminates in the central nervous system, and a peripheral process that extends from the sensory receptors. Then we have motor or efferent neurons, which carry impulses away from the central nervous system to effector organs. Most motor neurons are multipolar, and their cell bodies are within the central nervous system, and they will form junctions with all the effector cells. And then an interneuron will lie in between motor and sensory neurons, and this is always combined to the central nervous system, so we'll find neurons a lot of time in the spinal cord. So here's a look at multipolar, bipolar, unipolar, and where examples we would find them and how they work. Neuroglia, then, there are 6 types of neuroglia. And again, neuroglia are just the cells that support the neurons. There's 4 types in your central nervous system and 2 types in your peripheral nervous system. So the most of the neuroglial cells have branching processes in the central cell body. They outnumber neurons about 10 to 1, and they make up half the mass of your brain, and they can divide throughout life. Astrocytes, this is a good test question, are the most abundant glial cell type. They function to include regulating your neurotransmitter levels by increasing the rate of neurotransmitter uptake in regions of high neuronal activity. The signaling increases blood flow through the capillaries and active regions of the brain, and they help to control the ionic environment around neurons. They produce molecules necessary for neuronal growth like B, T, D, TF, and they also propagate calcium signals evolved with memory. So this is a look at what an astrocyte looks like. Here's the cell body with the nucleus. It has these feet that look like, they're called perivascular feet, and they wrap around capillaries to produce a barrier between the blood in the capillary and the brain tissue surrounding it. So we often called astrocytes that they make up the blood-brain barrier. Microglia are the smallest, and the least abundant glial cell. And again, we're only talking about neuroglia and the central nervous system. So astrocytes were number one, we're in microglial. These are like phagocytes. They're the macrophages of your central nervous system. They will engulf invading microorganisms and dead neurons. They're derived from blood cells called monocytes, and they'll migrate to your central nervous system during embryonic and fetal periods. So here we have the microglial cells. They're a type of defensive cell. So they help to engulf and get rid of anything foreign. Here are ependymal cells, which line the central cavity of the spinal cord and brain. They will have cilia, which helps circulate and transfer the cerebral spinal fluid through the different cavities of the brain. Oligodendrocytes have very few branches, and they wrap their cell processes around the axons in the central nervous system. And the thing about oligodendrocytes is that they produce myelin sheaths, but in the central nervous system. So here's what neuroglial cells look like. They have the cilia, which help to transmit the cerebral spinal fluid in its cavities. Here are what oligodendrocytes look like. They have myelin sheaths that wrap around axons to help axons transmit their signals faster. Then we'll get to the neuroglial cells in the peripheral nervous system. There's only two types. Satellite cells surround neuron cell bodies within the ganglia. And Schwann cells are also known as neurolemocytes. They surround axons in the peripheral nervous system, and they produce myelin sheath around the axons, but again in the peripheral nervous system. So here's a look at a satellite cell around the cell body, and then the Schwann cells producing the myelin around the axons in the peripheral nervous system. Myelin sheaths then are segmented structures composed of the lipoprotein myelin. They surround thicker axons, and they form like an insulating layer to prevent leakage of the electrical current. So the big thing about myelin is that they increase the speed of an impulse conduction down an axon. In the peripheral nervous system, myelin is formed by Schwann cells. It develops during fetal period and in the first year of postnatal life. Schwann cells will wrap in concentric layers, kind of like a lollipop, around the axon and cover the axon in a tightly packed coil of membranes. So here we have Schwann cells and how they mylate the neuron and how they will wrap themselves around the neuron to produce this insulated sheath to keep the action potential to travel faster down an axon. And here's an unmyelinated axon and how the axons don't have myelin around them. Actually, this was actually showing myelin around multiple axons, so just a different way that they're myelinated. The myelin sheath gaps then are also called nodes of Ranvier. They're gaps along the axon where there lacks myelin, and these will actually speed up nerve transmission. So because the action potential will jump between the myelin sheaths, thick axons are myelinated and thin axons are non-myelinated, so they will conduct impulses more slowly. Oligodendrocytes form the myelin sheaths in the central nervous system. They have multiple processes and they will coil around several different axons. Then we'll talk about gray and white matter in the central nervous system. Gray matter is gray colored, and it surrounds the hollow central cavities of your central nervous system. It forms a butterfly-shaped region, or sometimes an H letter, in the spinal cord. The dorsal, or the posterior half, contains cell bodies of interneurons, and the ventral or front half contains cell bodies of motor neurons. The gray matter is the site where neuron cell bodies are clustered. It's a mixture of neuron cell bodies, dendrites, unmyelinated neurons, and neuroglial, and it's where synapses occur, or junctions with other neurons occur. White matter then lies external to the gray matter of the central nervous system. It's composed of myelinated axons, and myelin is white, so that's what gives it its white color. And it consists of axons passing between specific regions of the central nervous system. Tracks are bundles of axons traveling to similar destinations within that white matter. Nerves then are cable-like organs in the peripheral nervous system. They consist of numerous axons wrapped tightly together in connective tissue. Most nerves contain both myelinated and non-myelinated sensory and motor axons, and the axon is always surrounded by those Schwann cells, which are creating the myelin in the peripheral nervous system. Then we have different connective tissue sheaths that surround nerves. The endoneurium is a layer of delicate connective tissue surrounding the axon. The perineurium is connective tissue surrounding a nerve fascicle, in which a nerve fascicle will be groups of axons bound into bundles. And then the epineurium is a whole nerve, and that is surrounded by the tough fibrous sheath of epineurium. So here we have, you can see the epineurium surrounding the entire nerve, and then around each fascicle, we have the perineurium, and we pull out one of the axons and myelin sheaths. Those are surrounded by endoneuriums. Here's a cross-section look through the spinal cord. We have the sensory afferent fibers coming in the posterior side or dorsal side of the spinal cord. So this is a sensory fiber. Here in black, we have an interneuron, and we have a motor or efferent fiber coming out the anterior or ventral side of the spinal cord. You can see here how the gray matter makes up this butterfly-shaped structure and how the white matter surrounds it, containing those different tracts. And then the central cavity is in the middle of the gray matter, and that will contain cerebral spinal fluid that travels down from the brain. So the central nervous system and peripheral nervous system are very functionally interrelated. The nerves of the PNS have afferent PNS fibers that respond to sensory stimuli, and then efferent PNS fibers will transmit any motor stimuli from your central nervous system to all the muscles and glands. The nerves of your central nervous system are composed on interneurons that receive sensory information, direct and transport that information to specific central nervous system regions in the brain and spinal cord, and then they will initiate the appropriate motor response. A reflex arc is the simple change of neurons that explain reflex behaviors. This determines the structural plan of the entire nervous system. And reflex arcs are responsible for reflexes, which are rapid autonomic motor responses that can be visceral, going to your organs, or somatic, going to your skeletal muscles. In a reflex arc, we always have the receptor, the site where the stimulus acts, the sensory neuron, which transmits afferent impulses to your central nervous system, the integration center, which consists of one or more synapses in the central nervous system, the motor neuron, which conducts the efferent or motor impulse from the integration center to the effector, and then the effector, whether that's a muscle or a gland. An effector can respond to effector impulses by contracting a muscle or secreting something from glands. So here's a look at components of a reflex arc. The receptor, for example, from a painful stimuli in the skin, sends that sensory information through the sensory neuron to the integration center. Maybe there's an interneuron there. And then the motor response travels out the motor neuron to the effector. So you can see here, all sensory information comes in the posterior side of the spinal cord or the dorsal side, and sensory or motor information then travels out the anterior or ventral side of the spinal cord. A monosynaptic reflex is the simplest of all reflexes. It's just one synapse. It's the fastest of all reflexes, is the knee-jerk reflex, and that's what your doctor tests when he hits your patellar or patellar tendon with a rubber hammer. So when he does that, he's testing your nervous system to make sure the reflexes are working. So pretty much automatically, as soon as he touches that patellar tendon, that stretch receptor sends up that signal. This motor signal comes out to kick up your leg as it extends it. The polysynaptic reflex is the more common type of reflex where one or more interneurons are part of the pathway. This is a withdrawal reflex, and interneurons will signal the motor neuron to contract the muscle involved. So an example of this is if you touch a painful tack, the motor response will tell your effector organ to lift up your arm. Then we can have different circuits, diverging, converging, or reverberating. Diverging circuits are when one presynaptic neuron synapses with several others. So that's called divergence. Converging is when many neurons synapse on a single postsynaptic neuron called convergence. And reverberating is when the circuit that receives feedback is via a collateral axon from a neuron in the circuit. I don't think I asked you guys many test questions about this, but these are different types of neuronal circuits. Then we can have serial processing and parallel processing. I'm going to pass over these as well because I don't think I talk about that. We'll talk a little bit about more integration. So for example, in a painful stimulus, how all of these networks come together and work between your peripheral and central nervous system. There's always an immediate response in the spinal reflex. Sensory information will still pass to the brain, and pain will be felt after the reflex is withdrawn. So let's say you burn your finger. To take your finger away from the point of pain, that doesn't involve your brain pathway. So it just goes into your spinal cord and the motor response comes right out. So this is a withdrawal reflex. It's extremely rapid. However, information is still sent up to your brain that pain was felt. Here's a cortical pathway, which is much slower, where simultaneously the nerve impulses travel on an axon branch that extends into the white matter, and this ascending axon carries the nerve impulse to the brain. So some disorders in the nervous system, multiple sclerosis is an autoimmune disease where the immune system attacks itself. It attacks the myelin around axons in the central nervous system. It varies widely in intensity among those affected. It's characterized by periods of relapse, disability, and remission or recovery. It affects people in different ways. For some reason, women are most affected, and it can sometimes affect women as early as 30, and its cause is incompletely understood at this time. Neural injuries may cause permanent dysfunction. If axons alone are destroyed, the cell bodies can often survive and the axons might regenerate. In the peripheral nervous system, usually macrophages will always invade and destroy an axon distal to the injury, so the axon filaments will grow peripherally from the injured site, and partial recovery is sometimes possible. In the central nervous system, Neuroglia never form bands to guide regrowing axons and may hinder axon growth with growth inhibiting chemicals. And there's really no effective regeneration after injury to the spinal cord in the brain. That's why when people go through, a spinal cord injury and are paralyzed, there's really no regeneration of nervous tissue to completely fix the problem. So here's an example. A site of nerve damage is if an axon is severed, there is a little bit of regrowth, some on cells, and there can be new myelin and sheath formation forming partially. So you can kind of see here regeneration of an axon. The nervous tissue throughout life, the nervous system develops from the dorsal ectoderm. I don't cover a lot of embryology in this course, so I kind of keep that out because embryology could almost be a whole separate course. So I'll end here with chapter 12. And so this takes us through the end of module 3. Thanks for listening, guys. Good luck on your next exams. You guys are doing a great job.